So semi-conservative replication of DNA, it was proved with the help of the Messelson and Stahl experiment. So what did they do is that in this experiment, they cultured the E. coli cells in the ammonium chloride medium. So they labeled them with the heavy isotope, that is the 15N. So 15N, it represents the heavy isotope of nitrogen. So that is the heavy isotope. So they labeled the first group of the E. coli cells, first group, with the heavy isotope, that is 15N. So the second group of the E. coli cells, they were labeled with the light isotope of nitrogen, that is 14N. So that became the second group of E. coli. So now what they did was that this E. coli that was, so the E. coli that was made to grow in the heavy isotope. So this was the both the strands of the E. coli DNA, they were inculcated with the heavy strands. So they were heavy. So this E. coli, then it was cultured on the 14N isotope of nitrogen, that is the light one. So what happened is that this is the first generation. So one generation of the E. coli, it takes place in 20 minutes. So E. coli, it takes 20 minutes to produce one generation of the E. coli cells. So here we have the parent heavy strands. So this is the heavy strand here, that is represented by the 15N, and this is also 15N. So now as they were made to grow in the light isotope, so they were made to grow in the light isotope, that was the light isotope of nitrogen, that was the 14N. So now it was observed that the light strand of the DNA was also produced, that was 14N, that were the new strands. So from this we were able to understand that DNA, it is able to replicate semi-conservatively. That is, it has the original parental strand, also it has a new strand, that is the light strand. So then they were further cultured in the light isotope of nitrogen, and then what it was found that we had the two, so we have here the two original parent strands. So as it is grown in the light isotope of nitrogen, so it will lead to the formation of the light isotopes itself, light strand itself. So here we also have the parental strand of the heavy isotope of nitrogen. Here we have the two strands also of the heavy strands, right? So we have the two strands, and because it is grown in the light isotope of nitrogen, the new strands that would be formed, it would be of the light strands, right? So in the second generation, that is after 40 minutes, we were able to find that there is equal proportion of the light strand and the hybrid strands. So hybrid, that consists of the both the light strand and the heavy strand, and the light strands, they were equal in the second generation. So this experiment, it proved that DNA has a semi-conservative mode of replication. So now let us move ahead to the next concept.